Hello students, now we are going to discuss meiosis. Earlier we had discussed the cell division process of mitosis. Now we are going to take in account meiosis. So there are few questions that we are going to deal with in this lesson. First is that the cellular division of meiosis, where does it take place? What are the cells which are destined to undergo meiosis? Then why is this meiosis? I had discussed earlier in the previous lecture that it is known as reductional division and mitosis is known as equational division. So why is this name given to meiosis, that something reduction would be taking place, something sort of reduction. So that is why it is known as reductional division, but we'll get to know that how that reduction takes place. Then we have a new term, that is a meiocyte. That why does a meiocyte undergo two divisions, why not one as in the case of mitosis? And there is another term that we face in this meiosis thing is inter-kinesis, one more term. So we have dealt with two types of kinesis, one was karyokinesis, other was cytokinesis, and now a new type of kinesis is there, that is inter-kinesis. So step by step we are going to see what all these things are. Now in question number 1 I had put to you that where does meiosis take place, and over here I am using the term meiocyte. So first of all, let's see what is a meiocyte. Meiocyte, cyte stands for cell, you must be knowing, and meo, something related to meiosis. So any cell which would undergo meiosis would be termed as the meiocyte. So basically meiocyte is the parent cell which has to undergo meiosis. Now what are the meiocytes basically? When we talk about meiosis, we are talking about sexual reproduction. When we talk about sexual reproduction, then we talk about the formation of gametes. See, meiosis would always take place when there is a story about reproduction, and which sort of reproduction? It won't be there in case of asexual reproduction. When two different parents would be involved, male and female parents, that time it would be sexual reproduction. And when we talk about this sort of reproduction, we do talk about gametes as well. So gametes are the cells which unite and fertilize and then give rise to a new cell which would be considered as offspring precursor. So basically the sexual reproduction is going to take place through gametes, and the cells which undergo gametogenesis would be termed as meiocytes. Now what is that gametogenesis? Gametogenesis is nothing else but meiosis. Two successive divisions which take place inside a cell and reduce its chromosome number to half. Now there is another thing that comes to your mind is reduce the chromosome number to half. So somehow I am reaching this term as well by using the sentence that the chromosome number is getting reduced to half. I am pointing towards the reductional property of this division. Now why do we need to reduce the chromosome number to half? The simple reason is that we need to maintain the ploidy level. So this is the purpose, basic purpose of meiosis. Along with this, there are other significant methods and procedures which are required for sexual reproduction and bringing in the variation in organisms that we see in sexually reproduced offsprings. Now let us see what meiosis is all about. Meiosis is all about, it is reductional division. There would be two successive divisions. One would be Meiosis One followed by Meiosis Two. Here I would deal with the last question which asks that what is interkinesis. Now when I am talking about M phase, basically mitosis or meiosis, I know it would be completed to its end by two processes. First being the karyokinesis, and it would be followed by cytokinesis. The nucleus would divide into two, then the cytoplasm would divide into two, and in the end of meiosis 1 we are going to obtain two daughter cells. Alright, these two daughter cells would have reduced their chromosome number into half. We'll see how it happens. But now these two daughter cells individually would go to the second division phase, that is meiosis 2. Then each cell would act as a parent cell for meiosis 2. These would further divide by the process of karyokinesis and cytokinesis into two daughter cells, and we will have 4 daughter cells as a result of meiosis 2. So the time or the phase basically that lies between this cytokinesis and the karyokinesis of meiosis 2, that is known as Inter-Kinesis. Alright, so to put it in simple terms, there is meiosis 1. When it gets completed, the time during which the cell stays before it gets into meiosis 2 would be known as Inter-Kinesis. The name only suggests that kinesis there would be division. It is the karyokinesis of meiosis 2. The inter means the time between the cytokinesis of meiosis 1 and the karyokinesis of meiosis 2 would be termed as Inter-Kinesis. Now let us take meiosis 1 and meiosis 2 individually. As it is M phase we had studied earlier, there would be 4 stages. So would be in case of meiosis. We have meiosis 1, which would have certainly stage number 1 would be prophase, followed by metaphase, third one is anaphase. I am talking about karyokinesis over here, that means nuclear division has to take place, and fourth is telophase. And because it is meiosis 1, so we put prophase 1, prophase, anaphase, metaphase and telophase 1 over here. So we get an idea that the division that we are talking about belongs to meiosis 1. Same is with meiosis 2, but over here what we need to learn is that prophase 1 is the most extensively active stage. Alright, this prophase 1, in this prophase 1, there are many things which are happening, many important biological phenomena like crossing over. We'll get to this, what it is, crossing over, recombination. These two things are very important when we talk about sexual reproduction, because these two things are going to lead to an important evolutionary phenomena that is known as variation development. You might not resemble your parents completely. There is a chance that two brown-eyed parents can give birth to a blue-eyed offspring. So all this occurs because recombination and crossing over has taken place during prophase 1 of meiosis. Now what is this prophase? Try to recall your knowledge of prophase, metaphase, anaphase and telophase from mitosis. We had simple division that in prophase the chromosomal content would condense, it would get detached from the nuclear membrane would dissolve. In metaphase there was formation of metaphasic plate. In anaphase there was separation of the centromere. Basically the centromere split and the sister chromatids got separated and they moved towards the poles, and in last it was reverse of prophase. This was what mitosis was all about. Meiosis would also be done in these 4 stages only, but the only difference that you need to remember is, please cram it by heart, it is homologous chromosomes which would come into scene in this particular division. Now you know, when I am talking about meiosis, in this case or wherever you would study cell division, basically to understand the cellular reproduction, you would take into account a diploid organism. A diploid organism is defined an organism which has 2 pair of alleles for expression of a specific character, and you know that alleles are present on chromosomes, so we have homologous chromosomes. I would define it in a simple way. Supposedly this is the chromosome and there is this part which is responsible for expression of X character. Consider it as X character. A similar chromosome would be present which would be exactly similar to this and would have a different allele for the same expression, that is X, in a diploid organism. So these chromosomes are homologous, that means the alleles happen to be located at same locus in those chromosomes. Don't confuse it with sister chromatids. Sister chromatid would be after S phase exactly similar material would be formed so that the newly formed cell gets the same. Please don't confuse homologous thing, that is alleles location, with sister chromatids. Now this happens to be a sister chromatid which would be copied after S phase, but the inherent ability of the chromosome to produce X character is present at this locus. So is present in this chromosome. So these happen to be homologous chromosomes. In case you don't understand it, get to know how a diploid organism expresses a particular character. There is always a gene which is responsible for expression of a character in an organism. That gene is present in two different forms on two different chromosomes. If the locus happens to be similar for different genes, then those chromosomes are known as homologous chromosomes. Now what meiosis is all about? It is all about bringing these homologous chromosomes together, then separating them by the first division, and then afterwards obtaining the division process in harmony with the other vital processes like crossing over and recombination. Now let us see how this prophase takes place. This prophase itself being a stage has further 5 stages. First being leptotene. I know these names are difficult to remember, but you will have to cram them by heart, followed by zygotene. When this zygotene is completed, there would be another stage that would be known as pachytene. Then there would be diplotene, and then there would be dikinesis. You can formulate any simple way to learn this L, Z, P, D, D. Anyways, you have to remember. So these are the 5 stages of the stage prophase of meiosis 1. These won't be visible in any other prophase, whether it is prophase 2 of meiosis 2 or whether it is prophase of mitosis. It is only during the meiosis 1 these 5 stages would be visible. Now what these 5 stages exclusively deal with is homologous chromosomes. So first of all, during the interphase, the cells, the meiocyte which has to undergo division, it would undergo its growth period where the synthesis of DNA would take place. That means the chromosomes which would enter the prophase would be like this, the original one and the sister chromatid joined at the centromere. This is what a chromosome would be as it enters the prophase 1. Now what happens during prophase 1, as this is taking place inside the nucleus? In prophase of meiosis, we simply said that nuclear membrane degenerates. So does the endomembrane system does. But in this stage, we are seeing the prophase as it is in further stages. So we are going to take in account the nuclear membrane, how it is present and when it dissolutes. So first of all, when we talk about leptotene, supposedly this is a cell. I would write these over here and make you understand it through a simple diagram. Supposedly this is a cell, alright. It is having a nucleus inside it, a huge nucleus supposedly. Now what happens, you know, prophase 1 is characterized by condensation of chromatin material. So few chromatins like this would come remaining attached to the nuclear membrane. They would arrange themselves like this and they somehow appear to be a single chromosome. Such is the stage of leptotene, where the homologous chromosomes are close to each other but they are not condensed completely. So we see it as a single chromosome which is attached to the nuclear membrane. That is the first stage known as leptotene. So what is the characteristic feature? That the homologous chromosomes are coming together, of course, and they are attached to the nuclear membrane and they are condensing, because we know that in prophase there is continuous condensation of the chromatin material. Now what happens next stage would be the stage of zygote. in zygote. Now this nuclear membrane would slowly start degenerating. So we will not take nuclear membrane into consideration over here. What we are going to discuss is those chromosomes which appeared as single, how they would change in this stage. So taking this as one chromosome and this as its homologue. See, I am considering the shape and structure, but I am taking a different color just to make you understand that these are homologous pairs. Whatever is being located over here would be located, it would have same locus for all the alleles. These two homologous chromosomes are going to come close to each other. So whenever I talk about zygote, please remember the term synapses, which is to be learned over here. Now what is synapses? Synapses is joining basically. These homologous chromosomes would come close to each other and join at specific points. Now take this as synaptonemal complex, which results in synapses. Now another term has come into being, that is synaptonemal complex. It would be proteins which would arrange in such a way so that the non-sister chromatids of homologous chromosome come close to each other and join like this. Alright, this blue part, if it is visible to you, this is a synapse as we call it. This is the joining part where the homologous chromosomes have joined to each other. And what are these? These are non-sister chromatids. Sister chromatid is this. If this and this is the copy, this would be the sister chromatid. These two are the sister chromatids. And because these two chromatids belong to different chromosomes of a homologous pair, these are non-sister chromatids which are interacting at a synaptonemal complex on the homologous pair. Now, if we see it under microscope, how it would look? You know, chromosome is basically chromatin material. This is one sister chromatid which is condensed like this, and this is the other one, another sister chromatin. So, how would be a synaptonemal complex present between? Of course, it would have like this. I am just telling you to make you understand that how does synapse form. There are lateral elements present to its side. There would be many places where this synaptonemal complex would be formed. The lateral elements join to the chromatin material, and there is central part. This particular thing is known as synaptonemal complex. Please pay attention. There are lateral elements which are attached to the chromatin material of the sister chromatids, and in between there is synaptonemal complex which joins the two sister chromatids. This synaptonemal complex, such synaptonemal complexes would be present all over the non-sister chromatids of the homologous chromosome, which would bind them. Now what happens next is that when the synaptonemal complex is being formed, they join to each other, and this is the characteristic feature. Now we are going to, the cell is going to transit to this